Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Kyle from Business Review, an NS media company, and I'll be your host today. It's our pleasure to have Ncardia with us, who's presenting this webinar entitled, A Paradigm Shift in Drug Discovery. So guest speakers are Stefan Braam PhD, CEO Ncardia, and Elena Matsa, PhD, Director of Discovery Technology. I'd like to welcome everyone to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link you received via email to rejoin the session. If you want to ask any questions, you can send those in via the questions widget. Just type them into the box at the top left-hand corner of your screen, click Submit, and we'll have some time at the very end to address any questions or thoughts that you may have. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But for now, please welcome Stefan Braam. Over to you, Stefan.
Thanks, Kyle, for introducing me. My name is Stefan Braam. I'm the co-founder, CEO, and CSO from Ncardia, and I'm happy to talk to you today about our drug discovery technologies. Ncardia is an iPSC technology company, and we are working with pharmaceutical companies in drug discovery and cell therapy. We help these companies to implement iPS technology with the ultimate aim to bring better medicines to patients faster. Ncardia is a worldwide operating stem cell solution and service provider. Our main operations are in Europe, in Leiden, the Netherlands, where we have our discovery technology and manufacturing technologies. And in Gosselies in Belgium, we have manufacturing technology and manufacturing operations. Our business development forces in Europe and US. We have 50 FTE, all dedicated to iPSC technology. We have more than 10 years of experience in the market, and approximately half of our people are PhDs working on manufacturing, drug screening, quality, product development, and sales and marketing. As a company, we are working in both drug discovery and cell therapy, which at the first glance might look very different to each other, but in fact there are big similarities between these two segments. So in every project we are doing, we are using iPSCs or other types of pluripotent stem cells. We manufacture differentiated cells at scale, and for today's topic, that's mostly cardio and neuronal. And then we do a lot of characterization and testing. So for example, to identify a molecule that is potent in these human cells. Obviously, we are also applying these same technologies in cell therapy, where the focus more is to develop a cell type that has potency in humans. And characterization and testing is there, not the molecule, but more the cell itself. But in terms of underlying technologies, the segments are very similar. You all know the classical drug discovery funnel, where a lot of molecules go in on the top and maybe 5 are selected for preclinical development. What is good today is that we see that almost every biopharmaceutical company is using iPSC technology to test the molecules preclinically to generate some kind of human proof of concept data. The focus of Ncardia is a little bit different. We focus on the top of the funnel because we believe that's the spot where the iPS technology can really have an impact. We can implement it there at a much larger scale, allowing for serendipity, allowing to identify molecules or targets that you wouldn't identify with more conventional technologies. So we are starting our projects with humans. We isolate skin cells or blood cells, reprogram them to iPSC. Then depending on the project needs, either repair a mutation, introduce a mutation, introduce a reporter, manufacture the differentiated cells at scale, and use them for drug testing. And what is very important is that this technology can be used with different modalities. So obviously with small molecules, but it's getting more needed or useful when it comes to other modalities like RNA therapeutics, where you want to have that human sequence proteins, antibodies, or in particular also in gene therapy projects. So I talked a few slides back about the drug discovery funnel and said that we as a company are focused on the top of the funnel to use the iPS technology where it's best suited. But in fact, when you look at it, you can use it at different stages, from target discovery up to preclinical development. So in target discovery, it can be used to validate a certain target, for example, by rescuing a phenotype, by modulating that target. It can be used for primary screening in a full phenotypic screen to identify molecules. It can be used for secondary screening. So for example, hit molecules out of a target-based primary screen. What is very interesting is to use it in hit-to-lead and lead optimization, for example, to establish SAR, structure-activity relationship, trying to identify a molecule that is both safe and potent and has no undesired side effects on the cells, up to preclinical development where iPSC from different patients can be used to identify which patient population is best suited for treatment with that molecule. And so these are different examples, and my colleague Elena Matsa will talk about that in her part of her presentation. So shifting gears a little bit to what Ncardia is doing in drug discovery. So what is very important is that iPSC-based drug discovery takes a lot of different technologies, and what Ncardia has been doing the last couple of years is bring them together under one roof by leveraging our 10 years of experience in this field. So what we try to do on this slide is to summarize all the different technologies needed, going from disease modeling to differentiation, manufacturing, and upscaling, to characterization of the cells coming out of that manufacturing process, up to assays that you do with those cells that might be upscaled towards screening. So on the disease modeling side, we are using CRISPR technology together with our partner Horizon Discovery to introduce mutations, to correct mutations. We are working with our preferred partners in universities and biobanks to source iPSC and license them in. And obviously we are confirming which mutations are in a cell. Regarding the manufacturing, we have established a variety of different manufacturing methodologies for cardiovascular and CNS cell types. It's important to say Ncardia is ISO 9001 certified, so we have ensured a very robust and reproducible manufacturing process. We are using a lot of different technologies, both for cell characterization and assay development, which range from electrophysiology to biomarkers to gene expression, signal transduction, etc., and I'll come back to that. We are trying to use as much as possible in our disease modeling processes phenotypic readouts, so to recapitulate the patient disease phenotype in the dish, think about how to make that, to increase the throughput and how to make that as predictable as possible and implement that if needed in a screening paradigm. So in terms of cell models, we rely a lot on established manufacturing processes, both in cardiovascular and CNS. So on the cardiovascular side, we have ventricular cardiomyocytes, we have atrial cardiomyocytes, we have endothelial cells and smooth muscle cells. On the neuro side, we have astrocytes. We have CNS neurons, which is a mixture of glutamatergic neurons and astrocytes. We are making peripheral neurons, cortical neurons, and microglia. And we are focused on organ systems because we see in a lot of projects that you want to be able to co-culture the most relevant cell types to model that disease accurately. So for example, on cardiovascular, it makes in a lot of projects a lot of sense to think about endothelial cells in combination with cardiomyocytes. On the neuro side, for example, astrocytes, microglia, and neurons. So after the manufacturing, it's very important to characterize those cells in the most appropriate assays. So what we are typically doing is to, per project, assess what we want those cells to do. So we start with our cardiomyocytes, our neurons, etc. and use a variety of techniques like gene expression, electrophysiology, morphology, signal transduction, maybe contractility or biomarkers to QC those cells. So already here it becomes very important that you have all those technologies under one roof. For example, in a safety project, you might want to be focused on specific ion channel blockers, while in another project, the signal transduction with gene X or Y is important. So per project, we assess what do we want the cells to do and QC the cells in that assay. After the initial characterization, we often do some assay development to develop an assay that really measures the disease phenotype and that is suitable for the rest of the project. So what we are showing here is that in terms of EFIS, we have different systems from different vendors to measure all relevant parameters from these cells in a reasonable throughput. We have different devices for impedance. In terms of gene expression, we do qPCR, RNA-seq, or sometimes even whole genome sequencing. We can do calcium transients at a higher throughput in either 96 or 384 well plate. We have in-house devices for contraction. We have different plate readers, for example, for luminescence and fluorescence assays that are often used, for example, to detect biomarkers. We have in-house high-content imaging with our image express confocal that can also be used in 3D. We're doing protein expression assays and metabolism assays. So basically, Ncardia is super well equipped to measure whatever is relevant in a certain disease phenotype. After establishing the assay, we also have capabilities to scale those up further. Already mentioned earlier that we have done primary screens up to 10,000 compounds. And here the goal is to try to get those assays as relevant as possible. So we use assay criteria to have, for example, a very clear link to the human disease. So for example, if contraction is important, we would like to see contraction in that high throughput assay. If a biomarker is important, we would like to see that biomarker. So it's very that translational thinking. We try to match that readout to make that as close as possible to the clinical readout. The key trick is then to make it high throughput compatible, which is the difficulty. And obviously there's a lot of focus to make it reproducible, to be able to screen with a low number of replicates, or ideally in an NS1, and still get good quality data. So to finalize this part and to summarize this part, where Ncardia can make a difference is by building better cell models that fully recapitulate key human disease features. By doing this in such a way that we can still screen, so that we have the throughput and we can help with stratification, so bring in iPSCs from different patients. So to repeat once more, we start with a patient, we build an iPSC-derived model for that disease. We use our large-scale manufacturing processes, ideally in a bioreactor, to scale the manufacturing to the appropriate cell number. We're developing appropriate assays and validate those with reference compounds, bring that to high throughput screening, and ideally identify molecules that have the potential to go back to patients. And with this, I'm happy to introduce to you Elena Matsa. Elena joined us earlier this year as Director of Discovery Technology and oversees all our drug discovery projects in cardio and CNS.
Thank you, Stefan. It's a pleasure to be presenting this webinar. I will begin by standing a little bit on the importance of selecting iPSC models for disease modeling and drug discovery. And I will use for this an excellent summary from a recent publication from Chris Denning's lab at the University of Nottingham. So what this outlines is the possible models for that can be used to model human cardiac disease. And ideally, we would like to use human cardiac tissue to model cardiac disease. However, availability of these tissues is limited and isolated tissues have very limited survival in culture. So they can typically withstand culture conditions for a couple of hours, after which the quality of the tissue quickly declines. This time may be sufficient to test a few compounds, followed by fixation and histology analysis. So this type of assay really doesn't render itself for high throughput screening and drug discovery. We can also use myofibrils from tissues and cells or purified actomycin fibers. However, these are really suitable for studies related to the cardiac muscle, the sarcomere, and do not reflect the entire physiology of a functioning human heart cell. Therefore, important phenotypes or side effects or mechanisms of action of a drug may be missed if such models are used. Animal models can also be used, and these are very good for showing systemic effects of medication. However, there are vast differences between animal models and human cells, for example, in important proteins for cardiac physiology, such as phospholamban, involved in calcium signaling, and also in certain myosin heavy chains. And this brings us to the need for having a human model that we can use to appropriately exhibit functional phenotypes of a disease in vitro. And also it brings us to iPSC-cardiomyocytes. We know that these cells respond appropriately to functional pharmacology, and they show appropriate disease phenotypes, and I will spend some time in subsequent slides going through these. And we know that in theory, in principle, iPSCs can proliferate indefinitely, therefore giving us an infinite source of cells that we can use for disease modeling and that render themselves to large-scale cardiac differentiation. So in the next slide, I will focus on the functional manifestations of disease phenotypes and also how at Ncardia we leverage the ability of cells to indefinitely divide in order to generate large scales of iPSC-derived cardiomyocytes. This table excellently outlines a summary of the current literature for iPSC-based disease modeling of cardiac disease. And it's not a table that I would like to go through in detail, but really the most important information here is on the columns to the left and the rows on the top. And I would like to use this table just to make the point that there has been really a large number of publications involving iPSC-based cardiac disease modeling since the discovery of iPSCs in 2007 and the Nobel Prize to Shinya Yamanaka in 2012 that range a wide variety of cardiac disease, from cardiac channelopathies and arrhythmia syndromes such as long QT syndromes to cardiomyopathies such as dilated and hypertrophic cardiomyopathy to metabolic diseases such as diabetes, structural abnormalities, and also diseases caused by pathogens such as Trypanosoma cruzi and Coxsackie virus. And that a variety of assays were used to characterize all these models. So important characteristics of cardiomyocytes can be studied using electrophysiology assays by multi-electrode arrays. Contractility can be assessed using impedance assays and video edge microscopy, calcium flux using calcium binding dyes, metabolic assays by monitoring the oxygen consumption rates of cardiomyocytes, and then the presence of several biomarkers and so on by high content imaging and also by plate readers. And so the second message that I would like to bring across with this slide is that really a well-trained and well-equipped lab is necessary for disease modeling, and that Ncardia has spent many years in developing these capabilities. As Stefan mentioned earlier, Ncardia has developed bioreactor manufacturing technologies to improve assay quality. So to provide a little bit more detail about this, we use medium and large scale bioreactors in order to generate up to 20 billion cells in a single batch. This is unprecedented and advantageous because it allows all assays within a discovery platform to be performed in the same batch of cells, therefore reducing any variation that may be caused, that may exist between different batches of cardiomyocytes. Briefly, we use a process that follows the development, the natural human development from pluripotency to mesoderm formation and cardiac specification, and then to cardiomyocyte differentiation, and store large batches of these cardiomyocytes that can be used for functional assays, anything from high throughput screens to secondary validation to SAR, and any safety assays that may follow. So what do we do after making all these cells? We discuss with clients what the best way to model their disease is. We help clients source disease, iPSC cardiomyocytes from patients, or perform genome editing in healthy donors. We also use our in-house iPSC lines to generate induced models of disease, meaning models that are induced either by drug treatment or pathogens. And I gave some examples earlier of Trypanosoma cruzi and Coxsackie virus, but COVID-19 would also be a good example of an induced disease model. Each of these possibilities of generating a disease model has advantages and disadvantages. Having a disease model that comes directly from patient-specific cells has the advantage of capturing the patient's genetic background. And most cardiovascular disease has either variable penetrance or contributing polymorphisms that account towards the disease severity. So capturing that genetic diversity and the genetic background that leads to the disease can be an advantage. However, especially for mutations that are family-specific or rare, there may be difficulties in recruiting the patient-specific cells. And this is where engineered models have an advantage, where there is a reduced need for patient recruitment. However, these models may be more time-consuming to generate. On average, an engineering project can take up to 18 weeks. And at the same time, there is a small risk for genomic instability, even though with current technologies, this risk is limited. Induced models also have the advantage of not needing additional patient recruitment and enabling the modeling of non-congenital conditions, as I mentioned. However, one should take into consideration that some of these conditions may not faithfully recapitulate the clinical disease. I will also spend some time talking about small details and things to consider based on our years of working with iPSC disease models. Upon receiving a new iPSC line, it's always good practice to check the quality, meaning the pluripotency, using very common iPSC markers such as OX4, NANOC, and surface markers such as TRA-181 and TRA-160, and also to check the karyotype stability. Once that's done, it's also good practice to keep cells at a low passage in order to avoid culture-induced genetic instability. Sometimes artificial culture environments can be harsh on the cells, leading to mutations that give them a survival advantage. And these don't happen often, but it's good practice to keep an eye on them and maintain a low passage number in order to avoid them. It's also advisable to test multiple iPSC clones from the same patient. This is because the reprogramming process is somewhat stochastic and can result in small differences in the pluripotency state of the cells, therefore leading to different characteristics in the differentiated cells and the disease model. So validating across multiple clones of the same iPSC line is a smart way to go. Using genetically matched control is also advisable. As I mentioned earlier, small variations in the genetic background can lead to either an increase or decrease of the disease severity. So using those appropriate controls is important. And if they're not available, using multiple unrelated controls is advised. This is because for any phenotype, there will be a range of readings between healthy patients and between diseased patients. Therefore, having those multiple healthy controls is important to identify appropriately any disease phenotypes. At the differentiated stage, it's also good to test drug effects in multiple clones or batches from the same iPSC clone. This is because during the differentiation process, there can also be some stochastic effects or slight differences in the purity of the batches that can again contribute to differences in the disease phenotype. And these are not expected to be big differences. However, it's good practice and solid research to always include multiple batches. Using a high number of technical replicates and independent experiments, I think this doesn't only apply to iPSC research, but any research, so I will not stand on this point any further. Assessing the differentiated cells at a consistent time point between different experiments is very important. As the cells are in culture for prolonged periods of time, their characteristics may change. So for consistency and easy interpretation of experiments, a consistent time point post-differentiation is important. Considering the effect of density when comparing iPSC derivatives is important. And here I will give the example of measuring contraction force in iPSC-cardiomyocytes, where a higher contraction force will be a result of higher seeding densities. So when comparing a healthy and disease line, you want to make sure that the result was because of the disease phenotype and not because of the seeding density. So these things need to be very rigorously optimized before the assay. Optimizing the assay's dynamic range, this would enable making sure that the phenotypes between healthy and disease lines have a big dynamic range between them and can allow for screening of drugs that improve the phenotypes very easily, assessing the assay window, so capturing that time point when the dynamic range is at its largest, and also specifying acceptance criteria and reproducibility of the assay is something that is good practice and that we typically perform with our clients. Finally, another good practice is optimizing cell culture media for each assay. And this is important and can be specific to the disease that is being studied. For example, for hypertrophic cardiomyopathy, we know that serum-containing media can inhibit the disease phenotype, so excluding serum is important when studying this disease. Additional examples include the exclusion of phenol red from media when assays that have readouts in fluorescence, absorbance, or luminescence are concerned, and also selecting appropriate media for electrophysiology assays such as thyroid media or other buffers. And in the next slide, I will give an example of the importance of cell culture media for electrophysiology assays. So here I describe a media optimization assay for cortical neuron electrophysiology with multi-electrode arrays or MEAs. Ncardia has a cortical neuron differentiation protocol that follows human development, meaning it initiates with ectoderm specification and then goes through multiple stages until functional cortical neurons are obtained. Our differentiations are very reproducible and lead to high purity cortical neurons, and the objective for this assay here was to identify a medium that enables maximum recording of electrical activity. 3 media were tested. The one that is typically recommended, medium A, here on the bottom panel of this slide, B and C, and you can clearly see from these heat maps that media B and C were much better than A, with C upon quantification identified as the medium that gave us spontaneous electrical activity in the majority of electrodes, And so these are the assays that are well optimized in-house and that we recommend to our clients. I will now describe some examples of disease modeling and drug-based assays performed at Ncardia. The first example is from a high-throughput screen used to identify compounds effective against hypertrophic cardiomyopathy. Hypertrophic cardiomyopathy is a disease clinically characterized by thickening of the heart muscle. It leads to disturbances of the heartbeat, which can manifest as arrhythmias, cardiac arrest, and even sudden death. They are usually caused by mutations in the sarcomere genes. In fact, 75% of cases are associated to sarcomeric mutations. And these devastating diseases really don't have a good clinical management at the moment. At Ncardia, we utilized 2 cell lines to model hypertrophic cardiomyopathy. One was from a healthy donor where cardiomyopathy was induced by endothelin-1, abbreviated here as ET1, and the other was from a patient with hypertrophic cardiomyopathy carrying a mutation in the myosin heavy chain 7 gene. For this disease, several phenotypes were assessed, including the cell size and, importantly, the secretion of a clinical marker known as natriuretic peptide B, or BNP as abbreviated here. Using our plate readers, we performed an ELISA assay to measure the BNP levels that are secreted by cells in the media, and identified that upon ET1 stimulation, or between control and myosin heavy chain 7 iPSC lines, there was a much higher level of NT-proBNP secreted. This gave us a dynamic range of 17, which is a very good dynamic range where against which we could assess the effectiveness of any drugs to reduce the proBNP levels, and therefore consider that as a surrogate for a disease amelioration. This assay is very amenable to high throughput screening, and therefore we wanted to develop it further in order to assess its robustness and reproducibility. We identified that it also had a very low signal to background ratio, a low % variability between controls, and a Z factor that was appropriate for high throughput screening. And here you can see some of the assay validation we performed before the high throughput screening. And we set up a 384 well plate full of test drugs, and as well as untreated samples and samples treated with endothelin-1 in the wild type iPSC cardiomyocytes. And we set up replicate plates of these experiments, plate 1 and plate 2, as you can see here. All of these selected drugs were tested at 3 different concentrations. And what you can see here is that between the 2 plates and across all concentrations and all drugs, there was a very good correlation. In fact, the Pearson correlation factor was over 0.9, confirming the reproducibility of this assay for high throughput screening. And you can really tell from the raw data here that this assay is suitable for selecting any drugs that either increase or decrease the secretion of BNP. In order to set up the high throughput screen using this assay, we performed a robotic platform that's set up in our facilities in Leiden. And this is a T-confluent workstation that has 3 robotic arms for pipetting and gripping. It is integrated with an incubator that can automatically store and host the cells, and also sits within a laminal flow cabinet. We had to rigorously optimize 3 protocols for plate coating with extracellular matrix, cell seeding, as well as medium refreshment and compound addition. The cells were maintained for several days in the robotic platform, at least 7 days, as you can see in the schematic at the bottom. And this included sitting on the 384-well plates, medium refreshments, compound addition for induction of hypertrophy in the wild-type line, and also compound addition from a library of approximately 5,000 compounds. And finally, harvesting supernatants in order to perform the BNP secretion ELISA assay that I described in the 2 previous slides. As a secondary screen, we also performed high throughput, high content imaging for BNP levels, and I will also discuss these in the next slide. Our primary screen was performed in 2 lines, the wild-type iPSCs and the myosin heavy chain 7 mutants. It included approximately 5,000 compounds from FDA-approved libraries, which were assayed twice, leading to approximately 10,000 data points being acquired over 30 plates. Initially, from the primary screen, 341 compounds were identified as hits, and these were validated in a secondary assay, which included the ELISA secretion of BNP as well as the high content imaging. From these, 192 hits were confirmed, and a more complete potency analysis was performed, including concentration response curves, which gave us very nice sigmoidal responses in the majority of cases, and also a very comparable EC50 between the healthy iPSC line where hypertrophy was induced with ET1 and the disease line that carried the myosin heavy chain 7 mutation. So from this, I hope that it's clear that Ncardia has developed a large-scale iPSC-based manufacturing platform to generate large batches of cardiomyocytes that can take assays from A to Z. We also have developed our Discovery screening platform, which can be applied to disease-relevant iPSC models and uses predictive, clinically relevant phenotypes. Finally, our cardiomyocyte disease-relevant phenotypes have been used to screen over 10,000 data points, and many of the confirmed hits have very drug-like properties, meaning sub-nanomolar potency, favorable DMPK properties, good safety profiles, and importantly, good biological relevance. The second example that I will describe is payload optimization for antibody-drug conjugates. In this example, a client approached us after developing drugs, and in this case, antibody-drug conjugates that were efficacious against cancer, in order for us to assess the side effects of these antibody-drug conjugates on the endothelial barrier. We at Ncardia can generate iPSC-derived endothelial cells in large scale, as for cardiomyocytes and neurons described earlier, and perform impedance assays to assess the health of the endothelial cells. Here, the high peaks and the consistently high endothelial barrier in the first diagram indicate a good health at baseline, and following compound treatments, especially at the high concentrations, anything above 100 picomolar, indicated a decrease in the endothelial barrier and therefore toxicity from compound here named as compound 12. In contrast, compound 13 did cause initial toxicity to the endothelial barrier, which however recovered as early as 4 days after compound treatment, and led to a full recovery 22 days later. Therefore, this kind of common side effect of antibody-drug conjugates, which is the local inflammation at the site of injection that you see clinically, is reflected here as a reduction in the endothelial barrier. But for compound 13, this was only transient, and it therefore enabled our client to select this compound for further clinical development based on the results obtained at Ncardia. In conclusion, Ncardia's iPSC-derived endothelial cells demonstrate good morphologies in culture and express high and uniform levels of endothelial markers. They form tight endothelial barriers, and we have dedicated equipment that can measure these as well as perform wound healing assays, in vitro angiogenesis assays, and sprouting assays. We have successfully applied these assays for compound testing and therefore have developed suitable in vitro vascular model systems for discovery, efficacy, and safety assays. The last example that I will provide is the development of an epilepsy disease model in our CNS product, which is a mixture of cortical neurons and astrocytes. Our CNS cells have functional glutamatergic and cholinergic receptors, and in this assay we have used the cholinergic receptor agonist carbachol, which is also known to induce seizures, in order to develop a model for epilepsy. So treatment of the CNS cells with 20 micromolar of carbachol resulted in increased firing rate as well as increased network bursts in the cells, and this is indicative of uncontrolled behavior that is reminiscent of seizures, and this can readily be used to model epilepsy in vitro. And this brings us to our last slide for today. And to really mention that at Ncardia, we value our relationship with customers. We, following initial inquiry, have a consultation with our clients and put them in contact with an expert scientific team to discuss the details of their needs and design a project plan that reflects the needs of the client. We put attention and time into fine-tuning the proposal, and after execution, we discuss the results with the client and carefully discuss the next steps. We value your feedback, and we want to make sure that our custom project covers your needs. So with that, I would like to thank you for your attention today. If you would like to know more about us, please visit our website at ncardia.com or email us using the address support@ncardia.com, and we'll be happy to take any questions.
Okay, thank you very much. It is now time for the Q&A. So for anyone who missed it, to take part, just type your question into the box in the top-hand corner of your screen. Type your question, click submit, and then wait to hear if it's read out now. I've had quite a few come in already, so we're just going to get started with. What makes screening at Ncardia unique? Why should we choose you instead of another facility?
Well, that's a great question. So at Ncardia, we have expertise in stem cell biology, particularly iPSC biology, and we have worked on projects with somatic stem cells such as myoblasts. And I have to say that these cells are particularly sensitive to the use of antibiotics. Their proliferation rate changes, and so our cultures and our environment are very clean. And we also have to maintain the cells in culture for a long time, more than 7 days in most cases. So because of this, we have developed the capacity to perform aseptic screening with long-term cultures. And we also have functional cell-based assays as endpoints. And this is in contrast to most screening facilities who use screening with short-term assays, maybe a couple of days. And they really cannot maintain cells in culture for a long time because the facilities don't have the cleanliness required. Also, most screening facilities are used to performing biochemical assays, meaning non-cell-based, and therefore they may not have the ability for functional endpoint screens. So Ncardia really stands out for their ability to perform assays that require long-term cultures in aseptic environments with functional cell-based assays.
Okay, very nice.
Yeah.
Okay, we've got another question asking, do you offer assay development services, and do you perform a risk analysis in your assays?
Yeah, of course. So in fact, we prefer our customers to think of us as a stem cell extension to their company, and we would love for them to come to us for their assay development needs. We develop custom assays to accommodate each project. We also perform a careful evaluation of the risks and identify mitigation plans where necessary. We include these as part of our proposals. And then we always get consent from the client before we implement any of the mitigation plans.
Okay, thank you. Someone here has asked, how do you account for the immaturity of hiPSC derivatives?
Of course, so this is a question that we get often, in fact, and at Ncardia, we make sure that we choose time points and media for our experiments that enable clinically relevant drug responses and disease phenotypes. So this doesn't mean that the maturation level of the cells is affected, but that we get responses that correlate to the clinical phenotype. We also, as a company, continuously monitor cutting-edge technologies that are available and become published by academic institutions, and we try to incorporate the cutting-edge technology in the iPSC maturation field into our services. So we always strive to provide the most functional assays.
Okay, thank you. How do you source your cell models, especially patient models?
We usually, we would like to think that we provide an A to Z service, meaning that we are very open and willing to help clients source cell models. So sometimes clients do come to us with their own cells. And if this doesn't happen, we also have a collaboration established with Horizon Discovery in order to develop disease models using genome editing, whether that's introducing a mutation or correcting a mutation. In addition to this, we use biorepositories such as eDisc and Coriell, as well as our key academic collaborators to source iPSC lines. So we like to think that we will find the way to source the cells that our clients need. In addition to this, we have significant expertise in developing and reviewing informed consent documents to ensure compliance with highest ethical standards. So hopefully our clients feel that they're in safe hands when it comes to recruiting cells for disease modeling.
Okay, very good. I think we've got time for one more question. So someone here has asked, you mostly talked about cardiovascular and neural disease models. Do you work in other disease areas?
Yeah, of course. And in fact, we always strive to extend our portfolio of disease areas. So we are always looking for opportunities to extend our capability in collaboration with customers. So we are open to any models that clients want to suggest.
Okay, very good. I'm afraid that's all the time we have for today, which just leaves a big thanks to both presenters for the great presentation, and of course to Ncardia for sponsoring this session. To everyone watching, you will be receiving an email shortly letting you know how to access the on-demand version of this webinar. Or you can always watch it at our website, which is www.business-review-webinars.com. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned. And follow us on Twitter at BRWebinars Daily Updates, and join our LinkedIn group, Business Review Webinars. Thank you all once again, and I hope everyone enjoys the rest of their day.